Hey all, it's Dr. B. This is going to be part two of the NCBI GEO gene expression um, tutorial for this course. So once again, you're going to go to genes and expression, and this time you're going to go to the data sets instead of the GEO database. And let's just throw in lung cancer. Let's say what we get when we hit lung cancer. All right. So here are a whole bunch of studies that were done, and you'll see this looks a bit like a microarray on the right. That's because it is. There's all kinds of versions of those, but this is some data. Let's just take a look at this one. So this is a study that was done. So let's just kind of click here real quick. What did they do? This is a no small cell or non-small cell lung cancer. Derived stem cell differentiation, where again they were looking at cancer stem cells and looking to see any sort of differential expression. There, it looks like there were 8 samples here that they had actually used. So what are we going to do? We're just going to click on this button called cluster analysis here, and this is basically called a heat map. And what this is doing is it's showing you the relative amount of expression of a whole bunch of genes and their relationship to each other. And you're like, this is overwhelming. Like, how do I read this? Well, the good news is all you have to do is click anywhere in here and you can move this bar around and you can see any area. So once I move it around, I double click it. And now I see that it's much more manageable. And what we have here at the top, these are the samples. So we saw this study had 8 samples. So there's 1, 2, 3, 4, 5, 6, 7, 8. So those are the 8 samples. They probably are coded based on a particular person. We also see a tree here based on the relative similarity between them and sequences and or expression. And then on the side here we have the actual genes. And so this is just all the genes from that little area that I looked at. And I can even zoom in more if I want. I can just kind of grab like this and then really get into detail. And now I can just look at these things. And so what does this mean? It basically means that if we take a look at this gene right here called ZCCHC11. Now you may be wondering, what the heck is ZCCHC11? So all I did is I just went back into NCBI and I typed in ZCCHC11 gene and then Homo sapiens. Here it is here. It's known by these many names. It is a terminal uridyl transferase 4, and this explains exactly what it does and where it is. So that's just a little side note to it. So let's go back and take a look at this particular thing. And what we see here is that in the samples, in sample 1, 4, 1, 5, and 6, this particular gene is upregulated, which means that the expression levels in it are high relative to a standard. So and to each other. And so that's just one small area. We can also go back and grab any other area we want. Like, let's see what's going on over here. And by the way, you can see how these are clustered too, which is really nice. And these can be organized in a whole bunch of different ways. You don't need to know that for this course. But here is a whole cluster of results which are really interesting, and it makes sense. You can see how with this tree these samples are more related to each other and these are more related to each other, and you can see that at least based partially on this data, how the greens are on the right and the reds on the left, at least for this particular part. But this would be really interesting if I was a scientist, if I wanted a bioinformaticist like you to compare these and tell me what things are of relative difference. And it would be very interesting to see what these samples are and why they differ the way that they do. And again, this could be something where these are controls and these are patients, but this doesn't really say what that is, and that's fine. But the point of this is to teach you an extension of the DNA microarray and to show you how we use that data and can interpret relative gene expression.